Okay, hi everyone. So my name is Anil Ori. I am a graduate student in Roel Ophaus Lab at the Center for Neurobehavioral Genetics at UCLA. My research interests lie in interpreting the functional dimensions from GWAS data of psychiatric diseases and understanding the mechanisms that underlie these traits. So the talk today will specifically be about a in vitro model system that we set up in the lab and how we evaluated this model system to be appropriate for the disease using a longitudinal experimental design and a polygenic disease risk from GWAS information. So just, so the outline, I included a couple slides in the beginning that are more clinical, just to share with you some underlying features of the disease itself and the architecture. I think it's important to understand, especially what the underlying architecture is, so we know what model system or models to apply when we want to study the genetics of schizophrenia. The second part will be about the results and the analysis we did, and the third part will be about the general framework we set up and the implications of if you want to use it for other traits, and also where I think this work could be valuable in follow-up studies. So on schizophrenia, it's a severe mental disorder that affects 21 million people worldwide. In the US, it's 2.2, so the point prevalence is about 0.7% of the population. So it is a highly heritable trait, but it's not completely heritable. So there are both genetic and non-genetic factors that affect the disease. It's a chronic disease in the sense that over 50% of the patients have long-term psychiatric symptoms, and 20% of them are chronic. Unemployment rate is very high. Life expectancy is reduced by 10 to 20 years. And in 2002, the estimate for the overall disease cost was over 60 billion. So up until now, there's no cure for the disease, and there's extreme stigma and discrimination. So understanding the biology that underlies this, we can help patients to get better, but also as a society, we can understand better what causes schizophrenia. So the clinical features, it's a slide with, I think the text is too small, so let me walk you through it. So I added this because I just wanted to emphasize the comorbidity between the diseases. So for example, if you look at schizophrenia, one of the core symptoms are delusions, hallucination, and there's also cognitive decline and social withdrawal. But there's also, for example, episodes of reduced moods or fluctuations in mood, and that overlaps, for example, with bipolar disorder. And bipolar disorder on its own has also, as an additional feature, delusions and hallucinations. So there's this comorbidity between the diseases, and there's also a genetic correlation between these traits. So I just wanted to emphasize this to keep in mind that that's the case. So I'll be talking a lot about schizophrenia, but clinically how these actually differentiate, it's up for debate. And especially genetically how these differentiate is up for debate. So majority of the psychiatric phenotypes are highly heritable. Age of onset of schizophrenia is around adolescence. So when you look at the developmental pathology of schizophrenia, I like this slide because it captures a lot of the theories that are currently out there. So on the x-axis, you see the developmental stages across life, where you have fertilization, birth, adolescence, and young adulthood. So the age of onset of schizophrenia is in red, which is around adolescence. And you see that there are genetic factors that are postulated to impact on during early development, but also in later in development, as well as non-genetic factors. And there are various biological processes that are related to brain function that are dynamic across development, and especially during in a phase that comes before the onset that changes a lot, where there's a prodromal phase where people think that there are symptoms that are already detectable for schizophrenia. So So when you look at the lifetime prevalence, like I said, so it's around 1%. Compared to other psychiatric diseases, it's one of the ones that at least it's not as prevalent, for example, as anxiety disorder or major depression. It's one of the most heritable ones. If you look at the twin-based heritability or family-based or from family-based studies. And so the heritability, you can also see it as in this plot where on the x-axis you see risk of developing schizophrenia dependent on the degree of relationship to an individual that has schizophrenia. So the more you share with an individual that has schizophrenia, the higher your chance of developing it yourself, which is captured by the heritability. So schizophrenia is a complex trait. This is a plot that shows the different, across the allelic spectrum, variants contribute to different effect sizes. So the GWAS, let's see where oh, shoot. Would you mind doing play again? Let's see, where is the pointer? Oh, got it. Okay, so GWASs focus mainly on common variants. And for schizophrenia, this has been one of the most successful GWASs for psychiatric traits. But there's also studies done on low frequency variants and more rare variants like copy number variation. But today, I really will focus on how we can use the information from GWAS to infer functional dimension of the disease. So this is a Manhattan plot, which shows on the x-axis the different chromosomes, and on the y-axis the association signal, the strength of the association signal for every SNP across the genome with schizophrenia. So this is the genome-wide threshold, significance threshold, and you can see that there are a lot of loci across the genome that are associated with schizophrenia. So there are 108. But there's also signal that is below the threshold. And so the SNP-based heritability, which is basically the aggregate of all these signals together, is around 23%. And we can leverage this information to learn about what underlies this signal. So for example, what people have done is, if you aggregate this signal across dimensions of genes that we know are specific to certain tissues, so CNS here is the central nervous system. What you see with schizophrenia is that it's highly enriched for genes that are active in the central nervous system, which is what we would expect because it's a brain disease. And if we zoom in even more, if you look at certain, so this is an annotation that is from a ChIP-seq mark that is a histone mark, and it's associated to active promoters. And it's from the fetal brain, and that's the most significant annotation that is enriched for the heritability of schizophrenia. And people have showed this before where there's this early developmental component that seems to be related to the genetic risk. So I added this just to show that, I mean, this is based on common variants, but if you look across the spectrum, although now it's still clear what the actual molecular mechanisms are, there is some converging evidence across the variants that we see that contribute when it comes to biological processes. So rare variants, there's been association, an overlap with developmental disorders, but also a neuron-specific signal that falls in the synapse. So I added this plot, particularly because the postsynaptic density, which is a structure at the postsynaptic terminal where genes are active that seem to be enriched for schizophrenia risk. Low frequency variants also show a signal for fetal brain, which overlaps with what we see in common variants. So we knew, so given this information, we were interested, well, okay, if you want to test, if the next step is like you identify these variants that are associated to your trait, ideally you want to have a system where you can actually introduce or you can test how these variants affect the cellular phenotype. We have limited access to the brain, so we need an in vitro model system, right? But then again, what model system to use? So we chose a model system that we wanted a model system that captured the in vivo context of the disease. So you need a cell type that's relevant for what you're interested in. Ideally across spatial temporal dimension of functioning that is related. And we thought that well, you want to ideally see overlap with genes from GWAS that are also important for this model system that you have. So we chose a system of human neural differentiation. This because it captures a specific molecular mechanism of early brain development, and we knew that the risk of schizophrenia up until now falls in neurons in the brain. And this was already an established protocol that was published on, where they showed an overlap in transcriptomic profiles with in vivo cortical development. So we thought this is a good starting point to test our framework of how to go about to evaluate this model system. So what I will show you here in the coming slides is I'll tell you something about the model system, functional characterization, and then there will be a part of how we went about in a data-driven way to identify pathways and genes that are active during the model, and in a longitudinal framework, and how we incorporate GWAS disease risk into that to say something about the enrichment of GWAS signal. Okay, and if at any point anything is unclear or you have questions, feel free to interrupt. Okay, so what we did is we used human neuronal stem cells. These are progenitor cells that have the capability of you can induce them to become any type of brain cell that you want. So we used an established protocol that you can induce them to become neurons. So across 30 days, we differentiated these stem cells to become mature neurons. At 7 time points, we collected gene expression data. This is array-based gene expression data. Oh. And so if you look at the first 2 principal components plotted against each other, you see that it captures the differentiation process nicely from day 0 all the way to day 30. So every time point was collected in at least triplicates. We wanted to have an extra confirmation that our model system was actually capturing neurons. So we went to a fact-sorted data set from the mouse brain where they fact-sorted 7 of the most common brain cell types. And we assessed genes that are specific to these cell types. And what you see here is that their average expression plotted over time, standardized to time 0. And what you see is that the average expression of about 50 genes that are specific to neurons seem to increase, but for all the other cell types like astrocytes, oligodendrocytes, microglia, they are not active in the model, or across differentiation, they are activated, which was a nice confirmation for us, and it's what we expected. So the next step was where we wanted, well, this is in vitro, and all this is happening in a dish, so we wanted to say something about the in vivo brain. And so the Allen Brain Institute has an atlas where they collected gene expression data of postmortem brain tissues of different developmental stages that you see here. So this is 4-week post-conception all the way down to birth and after. So what we then wanted to see, our test, is across the changes that happen across differentiation in a model, how does that overlap to changes that happen across the developmental station of the brain? So we did something that's called, we implemented a rank-rank hypergeometric overlap test. That basically, when you have 2 lists of differentially expressed genes, say 2 time points of your in vitro system, 2 time points of your in vivo system, you can just compare these lists against each other, what you see here in the matrix, in one matrix. And then if these lists would be completely identical, you would see a red line, meaning very significant p-values across the diagonal, which means that these have a really high overlap. So blue here means no significance, and red means that there's significance between these lists of differentially expressed genes between time points. So I partitioned the data set, the in vitro data set, in 3 comparisons. So the early time points, mid time points, and the late time points. And then we did this overlap test between all, in a serial way, between all the time points of the in vivo brain. And what you see is that as differentiation in vitro progresses, the overlap also progresses to later time points. Doesn't mean that we are actually capturing completely what's happening in the brain, but it's a confirmation for us that at least our differentiation process in vitro mimics and shows some signature of development that's happening in the in vivo brain. Okay, So that was some validation of the system and some functional characterization that we knew what we expected what was actually going on in the dish. So the next step was how to go about identifying differentially expressed genes across time. And then also, can we identify certain pathways that are active? So I chose to work with two methods that were published that use a Bayesian framework. And the reason why I chose these two is because they were explicitly designed to exploit the correlation structure between replicates and time points if you have them. So, and because we had at least 3 replicates, you can borrow information from the replicates to increase your power to detect differentially expressed genes. So I'll show you, I'll go in more detail in the second one, but the second one is an extension of the first one, which I'll tell you what exactly. So this method is called Bayesian estimation of temporal regulation, and it's a ranking method to identify differentially expressed genes. So what it assumes, I'm not sure if you can see this. So you have, say you have a treatment condition and you have a control condition, and you want to estimate the expression of a gene here, which is a function of the mean across all your samples that you have, and some error term. And they model the error term here as a multivariate normal that therefore captures the correlation structure between your replicates and your time points. And then so if you're interested in the difference between a treatment and a control situation, what you get is YG here, where you can jointly model these together, which will give you this multivariate normal with the delta G. And the delta G is basically what you're asking if you're interested in the difference between treatment and control, is you're basically interested in this difference here, and that's the delta G. So if there's no difference, that means that these two conditions are equal to each other. If there is a difference, you expect this model to happen. So what these authors propose is that we can basically model these two models given the data, and then we use Bayes' rule to ask the question, which of these models is more likely given the data that we see. And the nice thing, what they do, which is an extension from the first method, is they allow for this delta, the difference, to be a random factor. And they say that allows for a correlation to happen between time points, because you would expect that the delta here is more closely related to this delta here than, for example, this delta here. So they say that when you model that in this way, you will get increased sensitivity to detect genes that are sustained, they're differentially expressed really in a small amount, but sustained over time. So they want to increase power for those type of genes. So just to go in a little bit more detail, is that, so better is their function, and they compared it to like 3 other methods that are out there, and they showed that they had more power, and that especially the power lies when the genes is differentially expressed across consecutive time points, which you can see here. And the other method that is an extension of is this one here, which seems to have the most power at transient time points, when there's only one time when that's differentially expressed and it goes down again. So I use these methods in conjunction, and when you do that, we find that roughly the union of both methods. So first of all, these methods are highly correlated with each other, and that's because the changes in gene expression across differentiation are so large that I think any method would have detected these. They do show some subtle differences depending on the cutoff you take, but in the union there are roughly almost 8,000 probes that are differentially expressed, which map back to around roughly 5,000 genes. So and these genes are then the genes we call differentially expressed. And so the next step we wanted to do is like, can we then say something about certain pathways or correlation structure between these genes? So we applied a fussy C-means clustering mechanism, our method, that basically in Euclidean space clusters these genes based on a similarity in expression. So how does it look? So an example, so we identified 8 clusters, and what you see here is one of the clusters where you see the standardized expression on the y-axis and the differentiation across time with the highest members to this cluster, their expression level plotted with the mean in black. So if you throw these genes through like a Go annotation, you find, for example, that they are enriched for growth cone and polarized growth, neuronal differentiation, synapses and vesicles. So we apply cluster labels to these. So we call this the neuronal growth cluster. And we did that to all the other 8 clusters too. So this is what you see then. So we see 3 clusters that go down over time, which are cell cycle and RNA processing. There are 3 clusters that are upregulated over time, which are clusters related to neuronal functioning. And there's a transcription regulation cluster and an extracellular matrix cluster. So now that we've got more insight in what the biological mechanisms are that are active across differentiation, we wanted to go to the next step and then incorporate GWAS information into this. So how are these genes linked to the genes that we find associated to the GWAS of schizophrenia? So to do that, we went to 2 methods. So the first one is MAGMA, which allows you to do gene set analysis. And the second one is stratified LD score regression, which answers the question it answers a similar question, but in a different way, where you partition the heritability from GWAS across a specific gene set. While MAGMA, if we go into more detail, what MAGMA does is that it aggregates. So if these are your SNPs here, and these are individuals, and every SNP has an association signal from the GWAS associated with it. What you can do is you can aggregate these to on the gene level. So you basically compute a mean of the p-values per gene. And what you then can do is, here you have your gene IDs, here you have your mean gene association signal. And then say you have a gene set like neuronal growth, you can say, well, this gene is in this gene set, and these genes are out of my gene set. And what MAGMA does is basically it tests whether the average signal in being in the gene set is different than being outside of the gene set. And it's a regression framework. So it basically, so here it models, so G here is a factor of 1 and zeros, and you basically test whether this regression coefficient is higher than 0. And it's a regression framework that corrects for gene size, SNP density, and it also has a gene-gene correlation matrix to account for LD. Okay, so that's briefly on MAGMA. Stratified LD score regression. So this is a little bit more complicated, where stratified LD score regression is an extension of LD score regression. And this is a method, a cool method that was recently developed, where it uses an assumption where for a polygenic trait, if a SNP is an LD with a causal SNP, just because it's an LD, it will take over association signal with the actual causal SNP. So you would expect that that the SNP that you're looking at has a higher signal because of its LD with the causal SNP. And so it says that, so if you then plot the mean chi-square statistics across LD scores, you would expect a relationship like this for a polygenic trait. And what they then nicely showed is that if you then regress the chi-square statistics on the LD score, the slope of this line will be an estimate of the heritability of the trait. Now the extension of the method is that you basically do the same thing, but you do it in a subset of SNPs that are defined by your category of interest. So you can estimate the regression coefficient of a specific gene set that you're interested in, and you can ask the question, is it enriched for heritability of my trait? A caveat here that I want to note is that some SNPs, some categories in the genome are just, we know that they are just in general across all polygenic traits enriched for heritability. These are like, for example, non-coding regions or DNA hypersensitivity sites. So they account for a baseline model that they tested to give you a good estimate of enrichment that is above and beyond the background level of heritability which you would expect. So the results I'll show you, they're always accounted for this baseline model. Okay. So MAGMA is a regression framework that tests if the association signal in a set is higher than out of the set, and LD score regression partitions heritability across. So just to give you an idea of what I'm going to show you is, so here you have the gene ID. It's just a dummy example with the chromosome, the start and end site of the gene. This is so I'll use for MAGMA, because MAGMA it's easy to run with a continuous variable. I'll use the actual likelihood that we had from a gene having a chance of being differentially expressed over time. And in LD score regression, we'll use a dummy variable where we take in a cutoff of the most likely ones, and those are in the set of differentially expressed genes, and the rest is out of the set. And later on, so we'll first start with the genes that are differentially expressed, and then we'll move to if we have any signal, we'll move to the clusters. And then I'll do the same approach where I'll use the membership value as a continuous variable in Magma, and a dummy variable for LD score regression. So this is the two-step approach. So we'll first start and test the genes that are differentially expressed. If we find signal, we'll move to the clusters. So our interest was schizophrenia, but we've included several GWAS summary statistics from major psychiatric diseases that are published. We've used a negative control of Alzheimer's, that is a neurodegenerative late onset disease, and a non-brain phenotype that's human height. And then we use the 1,000 genome reference panel as an external reference panel to calculate LD from. So what you see here is the analysis on differential expression, on differentially expressed genes for Magma. So here's the regression coefficients from the model, and this is the p-value. And so for schizophrenia, we find a significantly higher association of the GWAS signal for genes that are differentially expressed. And for the other phenotypes, there's some suggestive signal here, but we didn't see much, and our negative controls did not show anything. So when you look at LD score regression, we see, so this is based on the dummy variable, not the continuous variable. So with schizophrenia, we find a nominal signal where there's an increased per-SNP heritability in the genes that are differentially expressed. So we continued with schizophrenia, and I'll show you results across the clusters because across both methods, they showed at least a suggestive signal. So when you look across the clusters, these are the 8 clusters, and this is the regression coefficients from Magma. So you see that the clusters that are upregulated, the association with schizophrenia seems to be distributed across them. And there seems to be depletion across the other ones. So if you do conditional analysis, because the memberships of these clusters, to a certain extent, they're correlated. So if you do a conditional analysis on each of these clusters while correcting for the others, the synaptic function cluster is the one that survives, and there seems to be some signal in the transcription regulation. We don't see anything for Alzheimer. But the GWAS of Alzheimer is also much less powered than schizophrenia, so that's something to keep in mind. Okay, so just to wrap up, I have like 3 or 4 more slides. So we were interested in investigating human neuronal differentiation as a model system for schizophrenia. We chose it because it overlapped with the in vivo context of the disease. It's a neuronal cell type, and it matches with the spatiotemporal dimension of cortical development. And what we now showed is that specific longitudinal gene clusters that are active across this model seems to be enriched for GWAS signal of schizophrenia. So it's a data-driven evaluation framework, and I think what I especially like about it is that we integrated the longitudinal transcriptomic signatures with polygenic disease risk, and that we highlighted specific clusters through that system. So if you then want to think about how does this work integrates with when we think about how to go from genomic findings to cellular phenotypes, this is how I think about it. So if you have this model here, where what we did is basically we brought these longitudinal genomic signatures in a regression model with polygenic disease risk, using the information from across the whole genome. So 2 things to note here is that this model system, ideally you want to see a relationship that shows a high correlation, but on both sides of the spectrum there's noise, right? There's the polygenic disease risk, which is basically the GWAS, which as sample size increase, the true signals will pop up more clearly. Here we have a very rough measure of some biological feature that's happening during neuronal differentiation, right? So if you look across differentially expressed genes, we see that it explains some of the variance. If you then focus on differentially expressed genes and you go to synaptic membership, we see that we do better and that the risk lies more in that. So the question is then, moving forward, this is interesting, but how specific is this to the disease? So how can we increase the variance explained in our genomic signatures? So what I'm really interested in is to taking this to the next level where, so we did a targeted approach now where based on evidence from literature, we focused on neurons. But ideally, you want to be unbiased and capture everything, every cell type that is present during development of the brain. So you could, for example, start off with differentiating these neuronal stem cells to different lineages. We focus on gene expression data, but you can integrate different levels of information. So protein expression or epigenetic variation or the 3D structure of the DNA. And I think integrating these levels of information across different cell type specific lineages, I think will give us more information and will help increase the accuracy in our signatures that will help us locate the disease signal. And then ideally, in that system that you then have, you can test, for example, specific variants that you find in the GWAS or rare variants you can find in other genetic studies. You can test them in the system in a biological setting that's highly relevant for your disease. Okay, so that's it. Some acknowledgement. So I'm in the lab of Roel Ophoff, and Luz, Meryl, and Remco have been very helpful in this project. So on the program, I added some of the papers of the methods that I've used on the longitudinal gene expression and especially the how to look for GWAS enrichment. If you have any questions or you want to learn more about them, feel free to message me. I'm happy to talk more about it. Okay, that's it. Thank you.
Let's take a 10-minute break.
Okay.